All right, so we are going to begin our discussion of stems today, and this is actually going to segue into a very long section on dendrochronology, which is how scientists go about dating trees. So let's start with the external form of a woody twig. We have leaf arrangement. You got a woody twig. It consists of an axis with attached leaves. They alternate between leaves that are attached to the twig or they're spirally going around the stem. Opposite means oppositely arranged. These are leaves that are attached in pairs. And whorled are leaves that are in groups of 3 or more. So here we have alternate, where you have a space. There's nothing directly on the other side of it. Opposite, if you have one leaf, there's basically a twin. And whorl is where they go all of the way around. Leaf attachment happens at a node, which is an area of the stem where a leaf or leaves are attached. An internode is going to be a stem region between the nodes. Blade is a flattened portion of the leaf, and the petiole is the stalk that attaches a leaf to a twig. Bud attachment. The axil is the angle between a petiole and a stem. Axillary bud is a bud located in the axial. It may become branches. It may contain tissues that will develop into a flower. And a terminal bud is a bud present at the tip of a twig, usually resembles an axillary bud but is often larger, makes a twig grow longer during the growing season. And then there are scars. These are bud scales. They are modified leaves that protect the bud. Bud scale scars are tiny scars around the twig that are left when the bud scales fall off of the terminal buds in the spring. Stipules are paired. They're leaf-like appendages that may remain throughout the life of the leaf. And stipule scars are tiny scars left when stipules fall off as buds expand in the spring. Leaf scars are tiny scars left behind after the leaves fall off. And bundle scars are scars left where vascular tissue that was connected to the leaf and stem broke off. The shape and size of the leaf scars and the arrangement of the number of bundle scars are characteristic of each species. One can identify a woody plant in its winter condition by means of looking at the scars and the buds. So we'll be doing an entire section on leaves after we finish the stems in the dendrochronology section. So the apical meristem is at the tip of each stem, and it contributes to an increase in the length of the stem. There's a dormant period before the growing season, and it's protected by bud scales and primadoria. Primadoria is a tiny embryonic leaf that will develop into a mature leaf after the bud scales fall off and when growth begins. When a bud begins to expand, the cells of the apical meristem undergo mitosis, which is just growth and repair, and 3 primary meristems will develop. The protoderm is the outermost of the primary meristems, and it gives rise to the epidermis. Then we have the procambium, which is a cylinder of strands to the interior of the protoderm, and it produces the primary xylem and phloem. The ground meristem produces 2 tissues composed of parenchyma scales, the pith and the cortex. And the pith and cortex in woody plants will eventually be crushed and replaced by new tissues from within. The bud primadoria, these are embryonic buds in the axils of the leaf primadoria that develop into mature leaves and buds. As leaves and buds develop from primadoria, there are traces or pieces of xylem and phloem branch off from the main cylinder. Leaf gaps or bud gaps, these are little thimble-shaped gaps in the cylinder of vascular tissue left by traces. Vascular cambium, we talked about this a little bit before. It forms from a narrow band of cells between the primary xylem and the primary phloem. Cells produced by the cambium, these are going to be your secondary xylem cells, your tracheids, your vessel elements, fibers, or other components from inside the meristem toward the center. Secondary phloem cells are going to be sieve tube members, companion cells, or other components outside the meristem toward the surface. And cells of the cambium continue to divide indefinitely. Phloem is cork cambium. There's a second cambium that arises near the surface of the stem. It produces cork cells, which are impregnated with suberin, and suberin is waxy and it's water resistant, which is why certain types of bottles will be plugged up with a cork. Keeps moisture from getting in there. So that is actual tree tissue. Cork tissues make up the outer bark of woody plants. It reduces water loss and it protects the stem against injury. Lenticles, these are fissures or small bumps on a stem that allow for gas exchange. Tissue patterns in stems. Steels. A steel is a central cylinder in the stem made up of primary xylem, primary phloem, and pith. Protostele consists of a solid core of conducting tissues. Phloem surrounds the xylem, and it's common in primitive plants. Siphonosteles are tubular with pith in the center, and they're common in ferns. And eusteles have vascular tissues in discrete bundles. They're found in most present-day flowering plants and conifers. Cotyledons. We talked about cotyledons in the last section with seed leaves. These store food that's needed for young seedlings until its first true leaves can start to produce food for themselves. So until photosynthesis can begin, basically. This is from flowering plants that develop from seeds. They usually have 1 or 2 cotyledons, and pines and other cone-bearing trees have several cotyledons, usually 8. Dicots versus monocots. We talked about this as well. So dicotyledons are flowering plants that develop from seeds, having 2 cotyledons, and we abbreviate that as dicots, and you know that that's not really known well in in taxonomy anymore. And monocotyledons still is a branch of taxonomy, and it's flowering plants that develop from seeds. They have a single cotyledon, and it's abbreviated as monocots. Now, your herbaceous dicotyledonous stems. Annual plants that go from seed to maturity within 1 growing season, so they die at the end of the season. These have green herbaceous non-woody stems. Most monocots are annuals, and some dicots are as well. Herbaceous dicot stems have vascular bundles arranged in a ring around the stem, and woody dicotyledonous stems. So the woody dicots have most of their secondary tissues arranged in concentric layers. Wood is considered a secondary xylem. It's the most conspicuous layer of tissue. It's produced seasonally in most trees, and growth takes place in the spring and the summer, which we'll talk a lot about when we do these labs. So spring wood is when the vascular cambium first becomes active in the spring. It produces a relatively large vessel of secondary xylem. And summer wood is produced after the spring wood. It has fewer vessel elements, and it has more tracheids, and it's usually going to be a little bit darker. The switch between the 2 types of growth, you have early spring and summer, produces the annual rings. So an annual ring is 1 year's worth of growth of xylem. It indicates the age of the tree. Each ring represents 1 year. It can show if climate and other conditions occurred during the tree's lifetime. Higher than normal rainfall, annual ring would be wider than usual due to increased growth. Caterpillars or locusts strip the leaves shortly after they appear. Annual ring growth will be narrow due to limited growth. If there's a fire, not resulting in the death of the tree, you may have a burn scar. So here is how they would determine the age. A scientist is going to use a borer to determine the tree's age instead of cutting it down. So if you want to look to see how old a tree is, you don't have to go in there and chop it off all the way. If you did, this is basically what you would see. But they're going to use a borer, and you'll see videos of this later on, and you can see the banding patterns right here. This is extremely tiny. It's less than the diameter of a pencil. It might be longer than a pencil, but it's extremely small, and it doesn't actually hurt the tree whenever they put it into it. You might think it does, but it actually doesn't. The tree will repair itself, and you won't even know that it was bored into. So there's different layers of the trunk. We have the vascular rays, which are lighter streaks or lines that can be seen radiating down from the center across annual rings when examining a cross-section of a tree trunk. Consists of parenchyma cells that may remain alive for 10 years. So you can see these are the vascular rays. The primary function is lateral conduction of nutrients and water from the stele to the cortex. Heartwood is older, darker wood toward the center. It ceases to function when the cells become plugged with tylosis. And tylose is outgrowths of parenchyma cells that dam up the vascular tissue and prevent further damage to the plant. It forms during stressful times or drought. The primary and basically only function is strength and support. A tree may live and function even after the heartwood has rotted and left the interior completely hollow. So if you see a tree out there that you can walk inside of, know that the heartwood has actually died out. Sapwood is younger, lighter, and it's functioning wood. It's close to the surface. It forms at roughly the same rate as heartwood, so there is always enough plumbing, so to speak, for vital conduction. So this is actually a living tree here, and it has the vital pieces that still function properly on the outer parts of it, and it was just hollowed out, so the heartwood has died. The heartwood and even part of the sapwood in these California redwoods and other tissues can be removed from a tree, and it's not really going to affect the tree much. Holes big enough for cars to drive through have been cut in giant coastal redwoods in California without killing the trees. Cone-bearing trees and conifers. The xylem consists primarily of tracheids. There's no fiber or vessel elements that are produced, and the wood feels softer. It's commonly referred to as a softwood tree. Wood of woody dicot trees is called hardwood trees. So pine might be more of a softwood, whereas oak would be a hardwood. Resin canals are scattered throughout the xylem and other tissues. A resin canal is a tube-like canal lined with specialized cells that secrete resin into their cavities. And resin is a substance that flows to wounds and hardens to seal the wound site. It can also be used as a defense mechanism. So whenever people would bore into a tree, it would be filled with resin. Bark. All tissues outside the cambium, including the phloem, are called bark. Inner bark consists of primary and secondary phloem. Outer bark consists of cork tissue and cork cambium. Older tissues composed of thin-walled cells become crushed and functionless, and they slough off. Mature bark may consist of alternating layers of crushed phloem and cork. Latex actually comes from plants. So latexifers are specialized cells or ducts resembling vessels that form extensive branch networks of latex-secreting cells. Latex is a thick fluid that is white, yellow, orange, or red, and consists of gums, proteins, sugars, oils, salts, alkaloidal drugs, enzymes, and other substances. Its function in the plants is not really clear, although some believe that it aids in closing up the wounds. Commercial uses for latex: rubber is the most important one. Chital tree produces a latex used in making chewing gum, and opium poppy produces latex containing important drugs like morphine or heroin. Other well-known latex producers are milkweed, dogbanes, and dandelions are actually latex producers. So that's kind of crazy, isn't it? Monocotyledonous stems. Most monocots are herbaceous plants that do not attain great size, like grasses and lilies. These stems don't have vascular cambium or cork cambium, and thus they produce no secondary vascular tissue or cork. Surfaces of the stems are covered by an epidermis. Xylem and phloem are in vascular bundles scattered throughout the stem, and bundles are surrounded by sclerenchyma cells. Sclerenchyma and thicker parenchyma cells are just beneath the surface, aid in withstanding stresses, so like big windstorms. Palm trees and other monocots. Palm trees differ from most monocots because they grow quite large as a result of their parenchyma cells continuing to divide. Other monocots develop a secondary meristem that produces parenchyma cells and secondary vascular bundles. Grasses have at the base of each internode intercalvary meristems that contribute to rapid increasing in length. And several commercially important cordage, like rope fibers, are obtained from monocots. Examples of these monocots are broomcorn, different types of hemps, and sisals. So that's what that looks like. And that is it for our stems. So we went through that a little bit quicker, basic stem tissues, but the big section here is going to be on our dendrochronology, and we'll be doing these labs over the course of many days.